Hello, everybody. I'm Tihana Kurtovic from University of Zagreb, and the title of my presentation is One Anti-Venom, Two Paths, which administration route wins against venom. And through the next few slides, I would like to give you not really an answer, but rather a new perspective on monitoring antivenom efficacy following intramuscular or IM and intravenous or IV administration. Snakebite and venoming represents a significant public health burden whose devastating impact would be reduced by the global assurance of safe and effective antivenoms. There is no standardized protocol for their administration across the Europe, similarly as in many other regions of the world. A recommendation has been made that antivenoms should preferably be given IV as a principle of harmonization of their pharmacokinetics with that of the target venom, since this should eliminate the restraint associated with the RM route. But until recently, almost all studies of venom and antivenom pharmacokinetics have been directed toward time measurements in the systemic circulation exclusively, and widespread belief that IV application is more effective than IM has been established. Accordingly, despite the lack of randomized controlled trials, it has been given a priority by authorities. However, there are a number of manufacturers whose products are still prescribed for IM use. But IM administration may not be such an incomprehensible concept if one considers that snake venoms are complex mixtures of proteins of variable molecular weight, which in most envenomation cases are injected into the interstitial space either by the IM or the SC route, and whose absorption into the bloodstream may occur by blood capillaries or small lymphatic vessels, depending on their size. Because of endothelial cells permeability, only peptides and proteins smaller than 10 to 15 kilodaltons can enter the blood directly, while others are being absorbed into the lymph. On the other hand, antivenoms, due to their uniform composition containing only large molecules, when administered by the same route, arrive at the central compartment by slow diffusion in the initial lymphatics, immediately reaching the venom coming from the injection site and timely preventing its escape into the systemic circulation. Specifically, there is evidence that the lymphatic system not only plays a role in venom distribution and bioavailability, but also serves as a compartment where antivenom extravasated from the blood after IV administration eliminates a large share of toxins before the lymph reaches the bloodstream. So the matching of venom and antivenom pharmacokinetics in the systemic circulation may not be the best or at least the only indicator of therapeutic success. The role of IM antivenom in the elimination of lymph-absorbed venom may be even greater, but it hasn't been studied, highlighting an important gap in existing research. A main objective was to compare two common administration routes in a comprehensive preclinical study involving FAB2 antivenom, specific for the venom of Vipera ammodites, a species of high medical importance in southeastern Europe, and determine which approach is more effective in overcoming either the whole venom or its neurotoxic fraction consisting of ammodotoxins in both relevant body compartments where neutralization occurs, blood and lymph. By gaining insight into the efficacy of venom elimination mediated by IM or IV antivenom separately in the blood and separately in the lymph. A more complete picture of the venom-antivenom interaction might be obtained, helping to identify the optimal route of drug delivery. A sheep employed as experimental model were envenomed by SE application of venom in a dose corresponding to a typical quantity yielded in milking of a mature snake. 2 hours after envenomation, they were treated with antivenom in an amount sufficient to neutralize the venom's lethal toxicity. Antivenom was administered by IM bolus in one group and by IV infusion in another. In each group, half of the animals, which were kept under general anesthesia throughout the experiment, were used for continuous lymph sampling by external drainage from ductus thoracicus into single tubes for 6 hours. The other half were used for a collection of blood at close intervals immediately after venom and antivenom injections, and then at decreasing intervals over a period of 2 weeks. In both blood and lymph samples, the concentrations of antivenom as well as those of venom and amoditoxins were determined by the respective in-house ELISA. The antivenom uptake into the lymphatic system was demonstrated, but differences were observed in the absorption rate and the total amount reaching the lymph depending on the administration route. The IM group experienced a faster arrival of a larger cumulative dose of antivenom over time. Compared to the IV approach, the absorption of IM antivenom was in average more than twice as fast. Also, its lymphatic exposure was in average almost 4 times higher. Because of better absorption properties, the IM antivenom was more successful in slowing down the influx of the venom and consequently reducing its cumulative absorbed dose. Both the absorption rate of the venom and its total amount reaching the lymph during the measurement period were generally lower when antivenom was given by the AM route. On the other hand, the absorption properties of amoditoxins were consistent in both groups, regardless of the treatment principle. More importantly, only the IM antivenom, even at a low dose, succeeded in achieving rapid and complete neutralization of the venom present in the lymphatic circulation. The IV antivenom mediated neutralization was only partial, and continuous venom absorption has been observed throughout the measurement period. Briefly, it appears that IM administration provides a more effective toxin clearance from the lymph. In the systemic circulation, both the IM and the IV antivenom show the typical pharmacokinetic profile. The maximum concentration of IM antivenom was significantly lower, and the time required to reach it was significantly longer in comparison to that given IV, possibly due to its prolonged arrival from the muscle tissue. Both approaches resulted in a similar elimination half time, volume of distribution, and clearance of the antivenom, as well as its total amount reaching the bloodstream during the measurement period. Although surprising, the phenomenon can be explained by the slow but sustained release of NAB2 fragments from the injection site, leading to the gradual accumulation and consequently comparable systemic exposure as after IV administration. But it appears that a sufficiently long sampling time may be required for the effect become evident. However, the IM antivenom resulted in a higher volume of distribution and clearance of the venom, indicating a greater propensity of toxins to leave the systemic circulation and enter the extravascular space, potentially decreasing the drug's neutralizing efficacy. But regardless of the treatment principle, neutralization of the venom occurred in a short period of time after the antivenom administration. It was followed by a pronounced secondary rise in its serum concentrations, likely because of delayed absorption from the peripheral tissues. The reappearance of the venom in the systemic circulation was much less pronounced when antivenom was administered by the IM route compared to the IV route, as clearly indicated by its, in average, 3 times lower systemic exposure measured during the post-treatment phase. Briefly, despite slower absorption and the lower bioavailability, the IM antivenom maintained therapeutic levels over an extended period and proved more effective in sustaining lower venom quantities in blood over time. The second objective was to obtain information on the efficacy of IM and IV antivenoms in terms of their ability to reverse disturbances affecting the main hematological and coagulation parameters in blood samples of sheep from the rescue study taken before and after envenoming and during the post-treatment phase. So that the administration route with the more pronounced impact on recovery successfulness might be revealed. All animals exhibited clear evidence of envenomation almost immediately after the venom injection. Hypofibrinogenemia, thrombocytopenia associated with reduced platelet aggregation, and leukocytosis develop progressively over time. The antivenom had a positive effect on the outcome, as the normal values of the laboratory were quickly restored after its administration, regardless of the route. And only the leukocyte count was not reversed by the antivenom and continued to increase, possibly due to the inflammation process or cytokine production. And final objective was to provide insight into the safety profile of IM and IV antivenoms. It was assessed by the evaluation of antibodies raised against equine FAB2 fragments as foreign proteins in serum samples of sheep from the rescue study collected 2 weeks after therapeutic intervention. The question was which treatment approach avoids or at least leads to a weaker humoral response, reducing the risk of late adverse reactions as a negative phenomenon associated with the antivenom use. But measurements of antivenom IgGs show that there was no significant difference between animals that received antivenom by the IM route compared to those that receive it by the IV route. And to conclude, for the first time, our study demonstrated the uptake of the IM antivenom into the lymphatic system, as well as its venom neutralizing activity in the lymph. It also enabled us to compare the impact of the IM and IV routes on venom clearance from both major body compartments where interaction with circulating toxins occurs. And the IM antivenom proved to be a good concept from the standpoint of preclinical assessment, especially if prolonged protection is required. However, it should be noted that its efficacy is currently applicable only to Vipera mollitus venom and not to snake venoms in general. And further studies with broader implications for other venom types and antibody formats are required to reach a definitive conclusion. Thank you for your attention. And if you're interested in more details, my colleague Erika is here with the poster and she will gladly answer all your questions. This study represents a major part of her doctoral thesis and I was just her supervisor.